Right, hello everyone. My name is Madison and I'm a researcher here at the NT Department of Primary Industries and Resources. This is the first of 9 webinars kicking off our NT Ag webinar series, held weekly every Friday afternoon, and we'll cover a range of different topics in these webinars. So, introducing today's speaker, we've got Darsh Rathnayake. He is an entomologist in our department here in the top end, and he's going to give a presentation on some cucumber green mottle mosaic virus bee research.
Thank you, Mary. So the presentation or the talk I'm going to present you today, it's one of the work that we are currently conducting in the Berrima Farm Science Precinct. So before I start the talk, I would like to say that you might not get any exciting results out from the presentation, but I would like to use this opportunity just to give you an introduction about our project now we are doing here in the Berrima Farm, and also to show you some results and the research update that we have done so far and what we have achieved. So, and hopefully, so stay tuned. Maybe next year, something like this seminar series, we might be able to give you like a full project presentation. Okay, so the CGMMV, since it's discovered here in the Northern Territory in 2014, the scientists here at Berrima Farm, they're working on many aspects of the CGMMV, or the cucumber green mottle mosaic virus. So the presentation or the project that I'm going to cover today, it's a kind of extension project from that big CGMMV project that we've been doing so far since its first incursion. So all these projects are funded by Hort Innovation Australia and the support of the Australian government. So if you really want to, so if you log into the HIA website, you can find out all the previous research and their findings and their experiments details and everything. Just log into the HIA website, or even the end of this presentation, I have given the link. So if you're interested to see what we have been doing, so you can just check it out as well. Okay, jump into the, just give me a second. And okay, all good. So yeah, so as we all call it as a CGMMV in the short form, it's actually cucumber green mottle mosaic virus. So this virus is the plant virus that infects most of the fruits and vegetables in family Cucurbitaceae. So that means most of the fruits and vegetables that we eat day to day are getting infected by this virus. That means watermelon, for example, watermelon, cucumber, zucchini, pumpkin, gherkin, they all get infected with this nasty virus. So once it's infected plant, as you can see here in these pictures, so leaves appear to have the mottle and mosaic pattern on the leaf, as well as on the later you will see the leaf yellowing. So if you check our infected fruit, as you can see here in this, sometimes flesh get rotten, looks like rotten fruit, as well as the mosaic pattern also in the fruits, not the even coloration. So these ones, once it get infected, not really good for consumption. So once you get a virus onto your plant, there is no any way that you can recover the plants, unless you just have to remove all the plants and fruits materials, or whatever the infected materials you have in the farm, or even sometimes you have to decontaminate everything that you have used in the farm. So that means basically this virus is contaminated or transmitted by a mechanical transmission, which is from any contaminated or the infected plant materials, or maybe from contaminated tools and machinery that you have used in the farm. So that means so you have to be very careful, and you have to follow most of the quarantine or the approaches to get rid from this virus and just further spreading through your farm. So other than the mechanical spread, there's a chance to get the virus through the infected seed. So if you have collected seed from infected fruit, so you may ended up getting some infected plant from the next generation too. So other than these two methods, so scientists have recently found out that there is a chance of getting this virus from the flower visiting insect, like such as the pollinator bees. So, but no one has really done so far looking at the behavior of the bees and its effect on the virus transfer. Let's say how many visits does it require, and what actually bee is doing on the flowers. Is there anything that specific bees might do on the flowers which enhance the transmission, like of thing, like those things no one has done so far. So for the history of the virus, so it's initially discovered in 1935 in England from cucumber plants. So since then, so it's spread throughout throughout the Europe, and then Middle East, and then Asia, and then year 2014, we got it here in Australia. So first discovery of the cucumber green mottle mosaic virus has occurred from the watermelon farm in Katherine here in the Northern Territory. Since then, so virus has been spreading all over the most part of the Western Australia, and also some part of the Queensland and some part of the Northern Territory. So now this virus is treated as endemic to Western Australia and Northern Territory, while it's still considered under quarantine control in other states, especially in Queensland and South Australia. Okay, what bees does in the watermelon? What's important of bees in the watermelon industry? So watermelon, as in many flowering plant, watermelon also require a vector to transfer pollen from male flower onto the female flower. So here in Australia, the main pollinator in watermelon is a honeybee, is a European honeybee. So not only honeybee visiting the watermelon flowers, so it's visited by many other pollinators and the visitors like native bees, ants, or sometimes butterflies I've seen on the flowers. So many visitors, but mainly the pollination it's done by European honeybee here in Australia. So study shows that to get a maximum fruit set or the high quality fruit set on the watermelon, if it's a seeded variety, at least its female flower requires 6 to 8 pollinator visits. So if you go for a seedless variety, it might be much bigger. So it says 16 to 24 visits it's required to get the maximum fruit quality, like a maximum quality fruit set or the maximum seed set. So basically, or simply, what does that say? That when you get the maximum, like when you increase the number of visits onto a female flower, there is chance to get more fruit or much higher fruit, like a much bigger fruits on the farm. So because of that, so watermelon industry practice keeping of honeybee hives around the watermelon farms to get more pollination done in the farm, and then also to increase the production or the yield in the farm. So CGMMV in bees. So can these bees, as I told you, so the main pollinator in the watermelon is bees, the honeybees. So can these bees transfer virus? So initially in 2014, the group of scientists have just trying to do it in the manual way, like a mechanical way. What they have done, so they got the male flower from the infected plant, and then it's rubbed onto a healthy female flower. And later they found that the healthy plant also got the virus. That they think that the same thing could happen from a pollinator, but in that case, it might be from mechanical as well. It's not really sure in that case now. So, but what we have done here in the last couple of years, so one of the field trial that we did, not really myself, the people who worked in the project previously, they have had a field trial where they put a positive plants and the negative plants with the beehive. So what they found when they tested the plants, they only could get the virus on the flowers, but not on the leaf. So they're suggesting that, okay, there is a vector or there is something going on with the pollinator. That pollinator can transfer virus onto the flowers. But so if we keep it a bit longer, we still don't know whether that transfer, the amount of the virus transfer onto the flower is enough for infection. So in that experiment. So that's mean, yeah, there's something going on with the pollinator and the virus. So in 2017, a group of scientists, I think in Israel, they have done a similar experiment in a cage experiment where they put a beehive and then with the positive and negative plants. And then at the end of the trial, they found some of the negative plants got infected too. So they're trying to omit all other pollinators or any other way of getting contaminated of their healthy plants. So they claim now, so it just basically because of moving around the healthy plants could got that result. That means those healthy plants got infected at the end. So what we think that could happen, so you have a healthy bee and then the bee visiting infected plant just to get pollen and nectar as usual. And then it's visiting a healthy plant in the farm. And then while it's collecting pollen and nectar from the healthy flower, they might deposit some virus on the flowers. And then the bees get infected and also later plants also get infected. So interestingly, so you might be surprised. So even I told you that the first CGMMV discovery happened in here in Catherine from a watermelon. But if you look at the literature, so the first detection of the virus has been from the honeybees. So similar period. So people who worked, I think Robert said that they found that they tested some bees from this area and then they detected CGMA from the bees even before biosecurity officers detecting from the watermelon farm. So it is interesting. So and the later people were interesting to see, okay, if a bee can have the virus, can the other bee products or the bee hives can have that in their the hives too. So Berima Farm researchers they found tested some bee products from those hives. They collected the broods, adult bees, pollen, and nectar, and everything that they could get out from the hive. And they tested it for CGMMV. Interestingly, they found that they have the virus in all of the products in the beehive. So not only they have the virus in the beehive, but also pollen, honey, and adult bee. So the virus that was in the pollen, honey, and adult bee was capable of infecting a healthy plant. That means they got the virus. The virus are the viable virus. That means they can reinfect a healthy plant. So other than that, they also found that the virus in the bees or the honeycomb could stay nearly 4 months or more than 4 months, and also those virus could be kept was able to infect a healthy plant. So that's quite scary when you look at the plant pathology aspects. So this is what could happen. So the bee collect the pollen and honey, and then while they collect those one, they get the virus and they take it to the beehive, and the beehive get infected, and that's what could happen. So these earlier findings suggest that even though you remove all of your infected plant materials from your field site, if you're sure there is no more any infection, there are no more any contaminated plant materials or machinery, but even if you have an infected hive, so there is a chance you get the virus back. So that means the beehive act as a reservoir for the CGMMV, even there is no any infected plants in the site or in the farm. So this is a big concern because the farmers tend to move the beehives around their farm and then just take it around for the next pollination reason. And time to time they move the beehives around just to get more pollination done. So if you take a contaminated beehive, there is a chance to get the virus back onto the healthy plants. So that's why we came up with this question: Can infected bees from the infected hive can transfer virus back onto the healthy plants? So we had this question. Along with this question, we had So this is what we don't know. So we have infected bee, and then can that bee infect a healthy plant? We're not really sure so far. So along with that research question, uh, we were granted funding to conduct a research project, 2-year research project. Along with that question, we also wanted to continue some of the research that we were doing here in the Berima farm. So just to test, if you remember, I told you that the virus can stay in the bee honey for nearly 4 months. What if how long it can be stays? Can it stay more than 4 months? Or maybe how about 6 months, 12 months? Can they still stable in the honey and then still be viable? We want to look at that one too. And what if you got an infected hive? Is there any way that we recover the infected hive just anyhow, because rather than destroying the beehive? So we want to look at that one too. Sure. And while we are conducting this experiment, collecting samples, we also want to know, okay, any other viruses that can be transferred by bees other than CGMMV. So we just going to look at other two common viruses that affect the watermelon: papaya ring spot virus and zucchini yellow mosaic virus. And also, we want to test what are the pollen or the plants that bees visiting in the farm site. Not only the watermelon, they might go for any other plant that they have around in the farm. So what are the other plants that they collect pollen from? So we are interested to look at that one too. Then we can just match that one with the CGMMV availability. So even though you don't have it in the watermelon, it might have in the weeds that are growing around the farm. So who knows? So as I told you, we were granted 2-year funding to conduct a research project to understand and manage the role of honeybees in CGME epidemiology. So this is funded by Hort Innovation under the Levy Melon Fund and with the help of the Australian government. So Brian Thistleton is a project leader, and the Mary, Maxine, myself, Shreya, and Vicky, we all work together to find the answers for the questions that I mentioned in the previous slides. So as I told you from the beginning of the talk, so I may not have exciting results to present you at this point, but I have some results because we started this project late last year. So we started with the field survey to test whether we still have the virus on the field site or the apiary in the Northern Territory. So we started with the field collection last year. So we sampled 7 apiaries in 5 in Catherine, 1 in each Darwin and the 10 Mataranga. So, and then we collected as much as we can, like a adult bee, brood, pollen, honey. We trying to collect all of the samples that we could get from the beehives. And then we tested those ones again using a molecular technique to detect any CGMMV. So what we found is interesting. So all of the apiaries, that means 5 apiaries sampled in Catherine, they got the virus in all apiaries. But in Darwin and Mataranga, we didn't see any virus on our samples. So, okay. So yeah, I told you. So we want to test the survivability of the virus in honey. So we know that it can survive for nearly 4 months, but we want to know how long it can be survived and how long it can be effective or viable in the honey. So what we have done, we collected some honey frames, honeycombs from the infected hives, and brought back into the Berrima farm here. And then we kept in a temperature control cabinet. We just wanted to keep it 37 Celsius. So we were collecting samples honey samples from each comb every month. So we are planning to collect it up to 12 months. That's mean at the 12 months interval, after the 12 months, we're going to test the presence and absence of the virus from the honey samples, as well as we want to try whether those viruses are capable of infecting healthy plants. So what if we don't get any infection or any virus availability at the 12 months? Then what we're going to do, we're going to come back to the sample that we isolated from the beginning. So we tested 11-month samples, 10-month samples, 9-month samples until we get the virus availability as well as their infectability. So what if we got the virus at 12-month samples and then they are viable? So we hoping to continue this experiment for another 6 months, but it's all depend on the fundings and resource availability in our project. So far we have collected honey samples for 8 months now. So our first testing will be in September this year. So fingers crossed, we hope that we will get there and then we get what we want. So yeah, can we recover infected hive? So is it possible to recover infected hive if we detect an infected hive? So just to find an answer for this one, just to try whether we can recover it or not, what we have done, like we done a pilot run, we just bought an infected hive from Catherine, and then we know that it's got infection. So those samples, hive, bee, and the bee, and the honey and everything was positive for the CGMMV test. So now we know it's infected. And then we brought it back here to Darwin, and we're trying to remove as much as the honey and pollen we could. And then we let bees refill the empty hive. And we kept this hive in somewhere where we know that there is no any CGMMV available, or there is no any CGMMV infection last couple of years. So we kept it there, and then we let the bees to refill the hive. And then we were collecting samples every day for a week since we removed the honey and pollen, and then every week for up to a month, and then we sampled it every month for up to 3 months. So maximum 3 months we gave them to refill and then to test. So unfortunately, all of the samples we collected so far from the beginning to the end, so they all came up as positive for the CGMMV test. But we have a feeling that even though we detect the CGMMV, maybe their amount or accumulation getting reduced over the time. But for the moment, we're not really sure what's going on. But we are planning to do more testing on this one just to test whether it's reducing, or even after 3 months, can those virus be infect, or can those virus infect another healthy plant? So that's in our future plan. Okay, so main questions: whether infected bees from the infected hive can transfer virus onto the healthy plants. So even previous studies showed that yes, bees can transfer virus onto the healthy plants. So not really sure how it's really happened. So how many bee visits it's required to get infected? If just a one bee visit from an infected plant would be enough to get the healthy plant to get infected, kind of thing. So we want to try the first field trial with the negative bees from the negative hive, and we having the positive plants and the negative plants same time. We're going to observe them throughout the day or maybe the morning hours, just to see how many bee visits it get from the positive plant to negative plants. And then we want to calculate, or we want to test how many bee visits it require to get infected. And also the flower availability, or whether it's so if the bee visiting a male flower or the female flower, could that difference of the flower male female could affect on the infectability, kind of thing. We want to test that one from our field trial, first field trial, which is we're currently doing. And once we got the answer for that one, then we jump into our main question, which is, can infected bees can transfer virus? So then the second field trial will be we using the infected beehive with a negative plant, and we're testing the same thing. How many visits does it require? Even a single visit from an infected bee onto the healthy plant, could that helpful for infecting a healthy plant? Also, depending on the flower type, like a male or female, does it have a difference of infectability, kind of thing. So currently we're doing the first field trial, but we're planning to do the second one soon after we finish this one, maybe early next year. So hopefully by same time next year, this time next year, maybe June, July next year, we might be able to find an answer for these questions. So other two small projects that we are doing in this project. So we want to test what are the other virus that bees can transfer. So we know that bees can transfer CGMMV, or they carry the CGMMV virus. Can they carry other virus? So the papaya ring spot virus and Sucking yellow mosaic virus, as I told you earlier. So these two viruses are quite infective for the watermelon. So we're going to test. So while we are collecting samples and testing it in the lab, so we also want to test it for these two potivirus types. So from the samples we collected last year, we tested. Even though we got the CGMMV virus from the samples, we didn't get any of these two viruses from our last year sampling. But we're going to do another sampling at the end of this year. So we hope to repeat the same thing again. So just to test, okay, maybe after this pollination season, they might get any other virus. Who knows? We'll see. And the last one, as I told you, we want to know what are the plants that are out there other than the watermelon or any other cucurbit plant. Is there any other the weeds that might have the virus, or weeds that might be visiting regularly? Or so we want to know what are the what are the plants that bees visiting, just to get an idea and just to inform the farm people, or you have these viruses around your farm, so you might need to be aware about that one, kind of thing. So what we're going to do, so all the pollen samples we're collecting from this field trial, from this experiment, we're going to use it. We're going to identify the plants that pollen belongs to using a next generation sequencing. So once we identified into individual species or genus level, then we can inform farmers just to be aware, or even we can get an idea. So because all samples we test for the CGMM first, and then send it for the pollen identification. Then we have a relationship with the presence and absence of the CGME versus the plants that we got in the pollen sample. So we hope at the end of this experiment or this project, we will be able to find the answers to the question that I mentioned here, and then that will be helpful for the melon industry in Australia. So as I told you from the beginning, so if you want to look at our previous CGMMV projects and their results, you can just log into their web link here, HIA website. So you can read all of the experiments and results we have done. And also, if you have questions, so you can ask me now, or you can send me an email. And that's it for today. But I will hope that I can give you a full overview of our project with the results maybe later next year. So I would like to thank all the people who are helping us to get these things done, and then also for the funding body, the Horticulture Innovation Australia and the Australia government. And then thank you all for your support and then listening.
All right, thank you everyone for participating, asking lots of questions, and joining us today. Thank you very much, Darsh. So next week we've got another presentation coming up on Friday at the same time. The next presentation will be by Dr. Warren Hunt on the topic of industrial hemp regulation. So please join us for that one as well. And then we've got a whole series of 7 more webinars coming up on Fridays after that.